Tanny, and I am the host of the Let's Talk SADS Live, the Canadian edition, and delighted to be here this morning. I'm coming to you from Vancouver, so still morning here, although we had a bit of frost or hail on the ground, so it's feeling very much like the rest of Canada today out here. But so I am a pediatric cardiologist and electrophysiologist that has the privilege of hosting these conversations, and they really are conversations where we come together to learn together about SADS conditions, sudden arrhythmia death syndromes, and many of you have been impacted directly. I'm going to share my slides. Huge thanks to Alice, Lara, and Aaron from the SADS US Foundation who help us put on these monthly webcasts. And of course, big thanks to our executive director, Pam Husband, who of course is the energy and the enthusiasm that keeps Canadian SADS going energetically here. So we're excited about our show today. It's really focused on research. And before we get to that, a bit of housekeeping and some announcements. We will be coming to you next month for our last show of the year with Melanie Kerr. And some of you will remember that we had Melanie on before with some really good Genetics 101 and a follow-up to Genetics 101, although many of us feel like we're still learning the language of genetics, which is, of course, in the last decade become so integral to managing and understanding SADS conditions. So it'd be great to have Melanie back. And then some of you will remember that last year we had some fun with trivia, and this year I've been sharing some fun facts as well. And so we are going to continue that tradition, and more on that to come. And a good opportunity to get involved and to maybe watch some of the archived ones, because you will need some memory of that. So head to the website and check out some of the old editions. We're very grateful to our sponsors, Lexio, Solid, Tenaya, and Thrive Therapeutics. Wonderful companies that really are visionary in their approach to SADS conditions, where they're changing the paradigm in how we manage. And some of you will have watched our conversation with Dr. Roxanne Carr, where we talked about medications and beta blockers. And these companies are really doing innovative things with new molecules and gene therapy, and really it's a paradigm shift in how we look after these conditions. So delighted that they are supporting us here today. And I always feel it's important to talk a bit about money. SADS, Canadian SADS Foundation, is a charitable organization and really depends on generosity of our supporters and some industry partners to do what we do, including me jabbering at you, but also some of the patient resources and keeping a website going, and really being able to be there as your Canadian advocacy group and partner in this SADS journey. So head to the website, maybe check out an old episode or two, and lots of great resources, and then click on the QR code and make a donation, however big or small you're feeling today. And on that note, we'll take a quick journey through the facts that we were sharing this year. We talked about catecholinergic polymorphic VT, CPVT. We talked about some historical things like arrhythmogenic cardiomyopathy and Brugada syndrome. We talked about long QT and how long we've actually known about it. We've known about it longer than we've been able to do ECGs, and the very peculiar association with hearing losses. As I mentioned, some of the companies are doing gene therapy now for SADS conditions, and gene therapy has been around a lot longer than we think. And then some really old studies, Galen talking about the autonomic nervous system, and we're working on getting a show about the autonomic nervous system for our 2025 lineup, because the autonomic nervous system is so important to SADS conditions and how we feel, how your morning exercise feels and goes. We talked a little bit about Wolff Parkinson White, and then last month, of course, we introduced propranolol and Sir Black, who invented the molecule. So today's fun facts is a special one, and some of you are going to recognize this young lady in this photo. The name is a little bit cut off, though, to make it hard. And so some of you probably know the answer, but I'm going to distract you for a moment. So here's a couple of things about 1995. So Bill Clinton spoke in the Canadian Parliament that year, and Labatt, one of our famous beer brewers, was sold to a Belgian company, and Chapters, the big book distributor, became incorporated in 1995. So the other thing that happened, though, is the Canadian SADS Foundation was established by Pam Husband and a few other parents, and she has been the steer at the head of this for a number of years, and really guiding us. So we owe a debt of gratitude to Pam that we have such a great group in Canada. So that's our fun fact, and I'm delighted today to have Dr. Michael Fridman join. And Dr. Fridman is one of the newer faces to the inherited arrhythmia scene in Canada, but really has a long background in SADS conditions. He's currently a pediatric cardiologist and electrophysiologist at the University of Calgary and works at the Alberta Children's Hospital there. We're going to bring him in to do more of his bio there. So let's please welcome Dr. Fridman. So, Dr. Fridman, thank you so much for joining me. I will occasionally slip into Mike and Michael, but it's great to have you here.
It's wonderful to be here, Dr. Sanatani. And I apologize if I slip into shoe every now and again.
Yeah,
I'll try my best to stay formal.
Yes, no one knows who Dr. Sanatani is. So, so give us a little bit of your journey here. You know, how did you get to be where you are now as one of the pediatric electrophysiologists at the Calgary group? So, where did your training take place? And
yeah. Of course. So the joke I always like to start off with is my father was an electrician, so I decided that I wanted to be an electrician as well, but I was terrible with power tools. So as I continued through my my educational journey, I started with an undergraduate at Queen's University in Kingston, Ontario, and found myself in an electrophysiology lab doing patch clamp technique on rat myocytes to try and understand the nature of the predominant sodium channel of the heart, and then continued doing a bit of research work in long QT syndrome, specifically, again, mostly on the patch clamp rig, and doing a lot of wet laboratory research. Once I had a bit of a taste of electrophysiology, I really couldn't get enough of it, and ended up in medical school, residency, and fellowships at the University of Toronto, where I did the bulk of my training at sick kids. And did a whole bunch of research there as well, trying to understand a bit more about other forms of SADS, like catecholaminergic polymorphic ventricular tachycardia, and trying to understand the clinical side a little bit more in a bit more detail. And eventually, as things transpired and grew, there was a position available at Calgary, and Dr. Robin Clegg, who is one of the electrophysiologists out here, who's been holding down the fort from a SADS perspective in southern Alberta, eastern BC, and western Saskatchewan for many years on her own. She was looking to have a partner in crime, and that's sort of how I came in, and really quickly joined the cardiogenetics group, and became one of the pediatric leads for the Southern Alberta cardiogenetics program. So it's been an incredibly fun journey and an exciting journey, and I'm incredibly grateful to all the families and the patients that I look after for keeping me humble and teaching me and learning with me and growing with me and putting their faith in me to be able to look after them.
Well, thank you. That's a great journey. And, I guess we have a few of those spots in common. I did my pediatric residency at Queen's, where our friend John Smythe was a wonderful inspiration and mentor. And of course, you also spent some time with our friend Bob Hamilton, who was also a guest. He gave us some great insights on our webcasts. I feel like it was last year already. Time is flying. So, well, thank you. And today we're going to tackle a big topic, research. And I want to start by being a bit nitpicky, as I tend to be, because the term research, I don't think is used the same by everybody. And we all have, families that come in and say, they're famous for saying, I did my research on Dr. Google or Wikipedia, and this is what I think. So clarify for us a bit what we're going to talk about in terms of research and what that field means to you.
For sure. So research, as everyone knows, is a pretty broad term where you're trying to find an answer to a question. And I think that's broadly how people understand research, in the sense that they have a question. For example, I have palpitations, or I fainted during exercise, or I have a relative with long QT syndrome. What does that mean? And finding sources that are reliable and consistent to be able to answer that question can sometimes be really challenging. But what we're talking about a little bit more today, and you and I share that sort of semantic nitpicky trait, I think. But when we're talking about research in the medical sense, it's really using a structured and standardized approach to asking a question and finding an answer to that question. So while a lot of people think about research as just answering a question, in biomedical research especially, research is more of a process, not just finding the question, but understanding the tools and techniques and the skills that are needed in order to unwrap that question and answer it as accurately as possible.
I think, you really hit on some key components there that I think are important. It really is that systematic and structured approach. And again, I don't want to spend too much time on this, but this whole, the quality of the information that you get. This morning, as I was driving in, there was a talk about our social media platforms, and we get so much of our information that hasn't necessarily gone through that rigorous vetting process. Whereas the research that we're going to talk about today really has a systematic process, and the data has been very carefully analyzed and curated. So let's talk a little bit about that sort of. So, for medical information to reach a place where it has been what we call peer reviewed, tell our audience a little bit about the steps that have gone on in the background to get to a peer reviewed publication.
Yeah, I think one of the things that always surprised me about the research process was how long it took and how many steps there were in order to make sure that not only was the question being asked in the correct way, but also that all of the tools, techniques, and tricks that are used in order to answer that question are part of this really long and drawn out process. So the general practice in research is to make sure that you have a good foundational element and a good base understanding of the literature that's out there in the scientific community. For example, reading all of the updated papers on long QT syndrome type 1, the KCNQ1 gene, knowing in great detail what do we already know about this gene from a laboratory and animal studies and cell-based trials, what epidemiologic or statistical information do we know about it, and what human trials have been done to try and understand what managements are able to work and not work. Because you don't want to end up in the trap of asking a question that's already been answered or already been disproven. Once you have your question and you've developed it from that understanding of the underlying literature, you then develop your hypothesis that you want to test, and that's the question that you're asking. So many years ago in the long QT registry, they asked the question, does the beta blocker that we choose matter in terms of the management and the protection of long QT? And once you have that question, you design the study in order to answer it. And for a condition like long QT syndrome, which is about 1 in 2,500 to 3,500 people, you can't just walk around on the street and ask for volunteers. You have to find a large population of people affected by the condition, or find a cell line in the wet lab, or a mouse model or a rat model, to try and understand the nature of the gene a little bit more. And once you have that process and protocol set up, you then have to apply to something called a research ethics board through a university. And a research ethics board is a group of people in the organization to make sure that every single research study that's done within the institution has a rigorous and ethical background to it. We cannot take advantage of people. We cannot negatively affect people. We cannot put them in harm's way in order to answer the question. Not that most protocols are doing it, but there are lots of sensitive pieces of information. So even questions as simple as, where are you storing the personal information of these individuals? Are you anonymizing? How are you anonymizing the data so it can't make its way back to the person who gave that information and participation for the study? Do you have all of the paperwork in place to explain things at a level that most people can understand it, so that they're not just being overwhelmed by the information and lots of scientific mumbo jumbo? So once that REB process goes through, and that can take months in and of itself, we then get approval to do the study and start the recruitment process. And the recruitment process for a clinical study or an epidemiologic study usually involves asking patients to join and provide their information. And this is something that I'm really passionate about, in the sense that no one should or can participate in research in a coerced way. You have to give your consent, and informed consent. And I know that Dr. Sanitani and I will go through that in a little bit, in order to make sure that you have a full understanding of what it is that you're getting into and what it is you're participating in. You have the ability to withdraw from a study at any time without consequence, and choosing not to participate in a study in no way will ever impact your care with your doctor. And I can't emphasize that enough. Choosing not to participate will never change how we treat you. It is just whether or not people want to participate. Once the consent process happens and people are recruited and join the study, provide their information, we gather the information that we're looking at to try and understand the details of the disease. For example, Dr. Sanitani, you have your CPVT registry, and we gather ECGs and clinical data from people who are willing and able to join that study. And then from the data that's collected, we then perform statistical analyses. And the statistical analyses on those data, whether that's animal models and wet lab work, epidemiologic studies, or head-to-head comparison clinical trials, all of those statistical analyses are how we determine whether or not there was actually a meaningful finding. Is there an effect? Is there something that happened? Or is there no difference whatsoever between the two groups? In that process, as an example, in that long QT study that was done from the long QT registry, they actually found that there was a benefit to long-acting non-selective beta blockers preferentially over selective beta blockers. So nadolol and propranolol being a preferred choice to atenolol, metoprolol, bisoprolol. And that's why Dr. Sanitani and I and our colleagues generally prescribe nadolol or propranolol for long QT syndrome, because the evidence showed that that was a bit better than the other beta blockers that we had available. And that's just to answer the question. Now, once we finished answering the question, we need to now disseminate the information and the research that we've discussed. So writing a paper, putting together poster presentations, presenting at conferences so that the greater scientific community can learn about the information that we've acquired. And most importantly, almost more important than anything else, they're able to scrutinize the literature, the research that we've done. And that is a fundamental part of the research process. Have we had enough people who are experts in the field picking apart and teasing every single detail to make sure that as a scientific community, we believe the results that are there before we can then use it effectively for patient management? And that process, as Dr. Sanitani already alluded to, is called peer review. when we submit an article to a journal for publication, we submit the article knowing that a panel of 2, 3, or more experts are also going to be looking through and scrutinizing that research, providing feedback and comments, asking us to do different analyses or adjustments to the paper in order to make sure that we are as close to as possible to truth. And that's the goal of research. How do we get from point A, which is having a question and knowing where we began, all the way to disseminating information that has been so thoroughly reviewed and scrutinized that the likelihood that something is being published that's untrue is as low as humanly possible, and is therefore as useful as possible to build that body of literature to circle all the way back and start at the beginning of that process again. So it's a long, drawn out process that can take months to years to accomplish. There are many, 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 many people that are involved in every single research project that's done. And it's a really powerful systematic tool to really make sure that we are getting as close to truth as possible. That's really the goal here. Can we find the truths that we need in order to help people like you?
That was an incredible outline and timeline of the research process. I think I'm going to refer our new trainees to this webcast to play that back, because to understand that process is so important, and especially the time that it takes, because as you know, we will have a trainee or a medical student who's sort of, well, I've got 6 weeks. What can I, what big problem can I solve? So you raised a lot of important points that I want to come back to, and let's start first with the ethics board and the consent. So I think, people often will wonder, well, why do I have to, read a 10-page, document, and why is my teenager getting a separate one? So tell us a bit about the whole, the purpose of the informed consent.
Yeah, so the purpose of informed consent is really the idea that when we are doing things with patients, and when we were making decisions with patients and families to treat a medical condition based on what's already known, there's always a consent process that goes through and an information process, because most of the time when I say, hey, I'd like to start you on a pill, people don't just look at me and say, okay, Dr. Fridman, yes, whatever pill you'd like. Although some people do. But even if they do say that, then I will still go through the process of what is the benefit that I'm expecting to get from this medication starting, like again, using the example of long QT syndrome and Nadolol. I want to start you on Nadolol because there's evidence that suggests that this decreases the risk of sudden cardiac death and arrhythmia syndromes and fainting. So because of that, the benefit, I think, is really important for patients who have long QT syndrome, especially types 1 and 2. Then there's the next step, which is what are the risks of taking this? So we know that the benefits of Nadolol are protecting yourself from cardiac arrest and cardiac arrhythmias, but the risks of Nadolol are things like fatigue. Blood pressure can be a little bit lower. Some people talk about vivid dreams or nightmares that happen. Some people feel really tired and sluggish on the medication. For younger kids, blood sugar can go a little bit low. So knowing what some of those side effects are can help make sure that a family who's thinking about taking the medication is able to understand, okay, now I know the good things from the medication. Now I know the bad things about the medication. I can make a decision as a parent about whether or not I think this is the right thing for my child. Or a teenager, I can make the right decision for myself if you're an old enough teenager who has the capacity to do that, to bring that process through. But the challenge is when we're doing research and we're asking a question, the reason we're asking the question is because we have a suspicion about what we think the answer might be, but we don't really know. So when we're asking someone to put themselves forward for a research study, we are asking them to put a bit of faith in us and some faith in the process, because we're trying to figure out what are the benefits, what are the risks beyond what we think might happen, and what is really going to happen when moving forward in that study. So the informed consent process is usually a really long document approved by the Research Ethics Board to make sure that in proper lay terminology, you understand the purpose of the study, who is involved, who to reach out to if you have questions or concerns, how do you withdraw your information, talking about how your information is stored, what the benefits we suppose are, what the risks we suppose are there any is there anything that's being put forward in the study? For example, if you have to come to BC Children's or Alberta Children's to do a test for us, like a stress test, are we going to be giving you a little bit of money to go and take the bus to and from, or is there a transit pass that's included as part of it to get to and from the study to not financially impact or harm a family? So going through all of that process is giving everyone as much information as we can, anticipating as many of the material risks and benefits as we can think about. Material risks being the things that are most likely to affect you. But it is a much more exhaustive process than the medical consent process, which is going through in a bit more of an abbreviated format, because we tend to know a little bit more about the outcomes from a medical process than a research process. And to answer the second question that you were asking, Dr. Sanitani, the second consent form, or the second form that is a bit more simplified and a bit more abbreviated, that's often what we would call an assent form, not a consent form. And the difference between them is that, other than the spelling, and consent is a few extra points in Scrabble compared to assent, consent is the formal legal process to actually go through all of the material risks and benefits in order to be able to do this at all. Assent is a bit more of a simplified process to explain enough of the information to someone with maybe less of an ability to understand. Let's say a younger child, a 7 or an 8-year-old. Yes, they can understand some of it, but they may not be able to understand everything. And because of that, the parents are the ones who are giving permission to do the study altogether. But the assent process is the idea that the kid's kind of agreeing, yeah, I actually do want to participate in this. Yes, I do want to do that stress test, because, we're not going to drag your kid onto the treadmill and force them to do it against their will. They want to have a bit of agency as well. And one of the things that I like to do, especially if blood work is part of a study, is the parents can give the permission about whether or not this blood work is going to be drawn, but we have to get permission from the kid to do it as well. But the choices that they get may be, okay, do you want it to come from the left side or the right, or left arm or the right arm? Do you have a preference as to which one you want to do, in order to make this easier for you and give them a little bit of control over the situation, even after the parents have given full informed consent to the study?
Yeah, no, that's a nice example. Um, One of the other things I want to touch on is this concept of the peer review, and just mostly to remind our audience that information does go public before it has been fully peer reviewed, especially when people are presenting at medical conferences. Often these are preliminary data. Sometimes the paper has already gone through the full scrutiny, but sometimes these are findings that have not gone through the full process. And now there's even a phenomenon where sometimes a paper will be submitted, and there are third party companies that then promote the findings in a so-called what's called preprint. And this was particularly difficult, for example, during COVID, when data was really coming at us fast and furious, and it was really hard to tell what had and hadn't gone through the rigorous process. I want to change directions a little bit, or dive further into, because we are talking about SADS, sudden arrhythmia death conditions, and you've done a great job of mentioning some very specific roles of research. But talk a bit in the broadest of terms. Why is research particularly important in SADS compared to sort of the standard practice or the more common conditions that are out there?
Yeah, absolutely. And just as a very brief side note, I think the point that you make about the speed at which information is available, and the accessibility of information and disinformation, is actually a really challenging thing for us to do as medical providers and as scientists, especially because, as everyone can hear, the research process is a very slow process, and there's a joke in the research community of things moving at the speed of science, which generally is very slow. So when people make a random claim on the internet, or anecdotally, or things like that, it can have a really wide-reaching effect, and lots of people can hear and believe that thing without a lot of scrutiny and a lot of that research coming through from that full peer review process. And in order to disprove that claim, it actually takes a really long time. So moving at the speed of science can sometimes be a very slow process, and it can be a bit frustrating as a researcher to have to deal with that. But it is important to remember that the process is meticulous for a reason. And to answer the question that you were asking more directly, Dr. Sanatani, getting involved with research is actually a fundamental important in these SADS, a fundamental import in these SADS conditions, because these conditions, by their very nature, are rare. The most common condition that we're dealing with in the SADS sphere, long QT and Brugada, we're looking at most estimates up to 1 in 500 people for Brugada in the adult world. In the pediatric world, we're looking at around 1 in 2,000-ish people. Long QT is 1 in 3,500. CPVT is 1 in 10,000. And some of these diseases are extremely rare. So the more people that we have coming in to provide that information and to provide their stories, the more powerful we can be in terms of the how specific and how precise the research can get. Because right now, in the beginning, long QT syndrome, as an example, it was just long QT syndrome. There was one known disease. We now know that long QT syndrome is at least 13 different diseases with 13 different genes, and the 13 different genes that behave in very different ways depending on how it's affected. In the cardiomyopathy sphere, there's a big difference between an arrhythmogenic cardiomyopathy mediated by PKP2 versus an arrhythmogenic cardiomyopathy mediated by DSP. So by understanding arrhythmogenic cardiomyopathy as a disease, we do have some information that we can get, but the more people who get involved with their unique variants and their unique families and their unique experiences may actually help fine-tune the data that we're looking at and find more specific answers and more specific details that can help those families in particular. So there is this really important symbiotic relationship. And what I mean is that families give of themselves their information, their research, their genetic materials with full informed consent, of course, and no consequences whatsoever for those who don't want to participate. There's no pressure. But that symbiosis is that by giving us that information in order to be able to work with it, we can then use it, get new answers, and that may eventually feed back to the families as well to be able to provide better outcomes, do things in a safer way, choosing, instead of having a transvenous internal cardiac defibrillator, having a subcutaneous, which is a bit of an easier procedure and doesn't have the same kind of risks in other situations. So there are lots of things that these research studies can do that provide hope to individual families and their ability to move things forward. Because the reality is, if it is a genetic condition, almost all the SADS conditions are, there are going to be people in the family that are affected now, and there are going to be people in the family that are affected in the future. So how is it possible that we can give a gift that helps to work on the next generation of people who are dealing with these conditions? And one of the ways to do that is to get involved with researchers and get involved with research and join some of the networks and registries that we have across Canada, the United States, and internationally to really understand these rare diseases.
Yeah, and I think you've really touched on a very important concept as well. And people are often surprised, especially with different, healthcare systems, that their Aunt Sally, that her information isn't readily available to us. So research is one of the tools that we have to be able to link families, because in the conventional doctor-patient relationship, that is the only, those are the only two parties to the information. And we cannot pick up the phone and say, hey, Aunt Sally, cousin Mike here, can you tell us everything about your history? So research allows us to do some of that, expanding families, and also finding other cases of these rare conditions. So really important. And so you've used the word registries a few times. Maybe just take a minute here to explain to our audience basically what are these registries, and they're why are we using them so much in you mentioned our CPVT one, which is partnered with the Amsterdam one. There's a long QT registry that's been going on a very long time. And then we, of course, in Canada have the Hearts in Rhythm, the HiRO registry, which is also a big national project.
Yeah, absolutely. Registries as a group are effectively I don't want to use the word repository because it's a big long word, but it's a place where we can store a lot of information about individuals with particular conditions to try and understand that individual's condition in the context of a larger population. So one of the big challenges in doing research in rare diseases like the SADS disorders is that the diseases are by their nature very, very uncommon. So to try and understand them, we need to have a large enough sample size, a large enough group of people that we have similar features of the condition in order to be able to understand if what we're seeing is true, or what sometimes you'll hear it referred to as signal, or not true, or something that you call noise. So trying to adjust the radio dial and all you're hearing is static, but eventually you get the radio dial, and maybe I'm aging myself with radio dials. I don't know if people are using that as much anymore. But you turn the radio dial and all you hear is static, but eventually if you keep turning that radio dial a little bit more, you'll start hearing music start to come through. And because I'm in Calgary, it's usually country music on our stations. But you continue turning and tuning that dial, and eventually you get to the point where the noise, that static in the back, comes down enough that all you hear is the music coming through. And that's what we're trying to accomplish with research, removing as much of that noise as possible to maximize the signal to detect what we need to detect to answer the question we're trying to ask. So thinking about things like the HiRO registry or the International Pediatric Brugada Registry, headed by Dr. Gonzalez Corsia out in McGill, or the CPVT registry, which is housed in Amsterdam and UBC. All of these registries are the idea that people from around the world who sign on and get their patients to consent to these processes, whether it's an international registry, national registry, or local registry, so that we can put all of those data points into the exact same database to be able to do really large analyses or big data analysis. And with artificial intelligence, or AI, being really big in the media these days, Andrew Cron and Dr. Sanitani over in UBC are really interested in using this HiRO registry data in these AI frameworks to try and see what can we do with the data that we have from the patients that we know, with the genes that we understand, to better diagnose these conditions earlier, find better treatments. Can we use AI to understand the molecular structure of a channel and have the machine put thousands or millions of small molecule combinations to see how can we get something to bind in just the right way to make this outcome better? And the only way we can do that is by having a large database of people. Looking at my clinic on its own, maybe as a cardiologist, I'll see how many thousand patients over the course of a career, maybe in the tens of thousands of patients over the course of a career, but that's one person looking at lots of people, only some of whom will have a particular gene. But if I take my experience and Dr. Sanitani's experience and the experience from the other electrophysiologists and heart rhythm specialists across the country and across the world, and we all put our information together, now all of a sudden we have this huge amount of data to pull from and be able to pick away at those questions of truth. And that's how we were able to understand Nadolol with long QT. That's how we were able to understand a bit more about the role of internal cardioverter defibrillators in CPVT. It's how we're able to better understand the differences between the types of arrhythmogenic cardiomyopathies, and the fact that now we know that it's not just a RVC, it's not just a right ventricular cardiomyopathy. There's a much wider, broader understanding of what the disease is, and it's all because of people like you folks who come and join us and help us with your information to move things forward. So registries are actually a really easy way for a lot of patients and families to get involved with research without having to do as much, because oftentimes it's just the tests that we're already doing for you as part of your routine maintenance and screening.
So for our audience out there, I'll ask you a pointed question. Do you think that research, the way it's conducted in Canada, is safe? Is it safe for our families to join?
Absolutely. I think the way that research is conducted in Canada, we have amongst the most strict research ethics boards out there, and Health Canada is also very, very tight in terms of their regulation. In terms, I recently had a conversation with a Health Canada expert. Incidentally, because I was asking about the process for drug approvals and things like that, and what it would look like, because some of my families had questions about why is it that some medications are available in the US and Europe, but we don't necessarily have those medications in Canada. And Canada actually has some of the strictest and most tight regulations paired alongside Australia in terms of actually making sure that the manufacturing processes are really, really, really rigorous. And that same sort of rigor from the government side has actually applied itself to the research side as well, where the academic standards in order to do things are actually very, very tight. The universities themselves often have little variabilities in terms of how their research ethics boards will operate, and some will have a little bit more latitude than others in certain areas. But in general, research is very, very safe. We are not allowed to do research studies on people that is likely to cause harm. For example, we know that exercise can be dangerous in some of the SADS conditions. We can't just have somebody stop all of their medications and go sprinting back and forth and have a cardiac arrest to see how many shocks it takes to bring them back. That is absolutely not allowed, and under no circumstances would a research ethics board approve that study. But if there's a new molecule or a new beta blocker or a new antiarrhythmic that we want to try, for example, some people may be on flecainide, which is a sodium channel blocker that we can use in CPVT for patients who beta blockers aren't enough to keep the rhythm under control. There are studies that are looking at the effectiveness and efficacy of flecainide, and we know from certain experiences and certain lab studies that this is probably helpful. And now with the new research that's coming out, it is actually showing that for people who have harder to control CPVT, flecainide can be a very useful agent. But there is a risk to doing those studies and doing those side-by-side comparisons and trying to understand how is this drug going to affect this condition when we don't know 100% how it's going to work in people. One of the things that's really in vogue right now, as far as research goes, is gene therapy, and looking at using specially modified viruses in order to deliver gene editing technology into human beings. It's a little bit tricky to get that underway because it's not something that's been globally done before. But if we have some good studies like some of the ones that have come out of the US looking at gene therapy in rats and rabbits, and we see that, oh, well, actually, we have been able to see really major improvements in the rats with PKP2 arrhythmogenic cardiomyopathy, or the rabbits with KCNQ1 long QT syndrome. All of a sudden we have built this evidence base to say, yes, well, we've tested this molecule, we've tested this gene therapy really rigorously in this animal model. We know roughly what we can expect. Now, when it's time to move that into humans, what are the steps that we need to take to do this very safely? And there are many, many steps from a clinical trial perspective to very first and foremost establish safety and effectiveness. Number two, in a small population, figure out what dose is going to be effective. That's the phase 1 trial or a phase 2 trial you'll have heard. And only after all of those safety studies have been done is a phase 3 clinical trial, which is the before it becomes an actual usable therapy in the general population, where we need to have hundreds or thousands of people with a condition being treated to try and see, is there a difference between people treated or people on conventional therapy? So because of all of those steps that are built in and put in place, I do believe very strongly as a researcher, I wouldn't do it if I thought it was harmful, but I do believe very strongly at Research in Canada, it is very, very safe.
All right, great. Well, that was a very fulsome answer with lots of rationale as to why, you and me and most of the inherited arrhythmia docs in Canada are participating actively in research. I just want to, as our time is running out, I want to speak to one of the comments that one of our audience has raised, saying, as a Brugada long QT patient, I thought getting involved in a research project would be easy, but that has not been the case. So, I'm sorry to hear about that experience. I think it really comes down to, are you being looked after in one of the centers that is sort of a recognized, part of the Canadian network of expertise in in these inherited arrhythmia conditions, and we've talked about the HiRO network, and the SADS website is another good resource. And SADS in general is a great place to say, to make sure you are plugged in. So and there are a lot of people that are very capable clinicians who are not SADS experts. So it's never an insult to be to get, as a physician, to be asked. You say, are you the person that I need to be seeing? So we're at time here. This has been an incredibly informative discussion. I think we've given our audience lots of behind the scenes understanding why they get presented with that 10-page consent form, and why we really depend on them to then in turn bring them the data and then the improved outcomes. The hope, as Pam, our leader, is always reminding me, research really is the hope. And we just briefly touched on some of the exciting new things. We're going to have another session on those emerging therapies for sure.
Absolutely.
Michael, it's been an absolute delight to talk to you this morning. And I thank you for really, educating our audience and me. I always learn, I think, more than I anticipate from these talks.
No, it's been an absolute pleasure being here, and I hope that the audience found it informative and helpful. And as Dr. Sanitani said, ask about whether or not your doctor is involved in research. Ask how you can get involved, and even if they're not directly involved, because as I said, the process can take a long time, and even getting through the REB here in Calgary is it's taking a little while in order for me to get the approvals that I need in order to start recruiting patients. But if that's the case and they're having trouble, we can always send your information to someone who has the capacity to get you into those registries or talk to you about some of the research. So ask your cardiologist what they're involved in. Ask how you can be involved in research, and we're all very helpful. We're all very happy to send your information to some of our colleagues who are a little bit more plugged in to be able to get that underway. So if you had a hard time getting through, I'm sorry to hear that. But the HiRO network is always looking. The Pediatric Brugada registry is always looking. The CPVT registry and the Long QT registries are always looking, and we're always happy to plug you in. But it's been an absolute pleasure being involved. I was really happy to be here, and I hope to be able to come back and do this again sometime.
All right. Thank you so much. And yeah, such an important part of what we do is that systematic approach to asking questions. I was going to ask Dr. Fridman, but we ran out of time. I was going to ask him if there are any bad research studies, but maybe we'll save that. We'll save that for another time. Well, just to, as the year is winding down, we do have one more episode next month. December 17th, we'll be having Melanie Kerr back to talk to us again. And she's going to talk to us about genetics, which has become an essential part to understanding. As I mentioned, head to the Canadian SADS website and maybe watch some old webcasts, maybe brush up on the fun facts, because people may not know. Last year we gave away, I think it was $200 to the winner of the trivia contest. So we're working on, we're looking through the couch cushions to find a prize, and we'll look forward to giving you more information about that. As always, it's been an absolute privilege to be here with you, and I will look forward to next time.